Okay, welcome back. Now I've resumed the recording, and we are starting with the effect of microplastic block now. And I would like to welcome Martin Thiel, that's here with us today. Let me just briefly find my notes. Martin Thiel is a marine biologist with strong interest in the behavioral ecology of marine invertebrates and the functioning of marine communities. The large amount of floating marine litter encountered during their research and the complete lack of scientific information about marine litter from the Southeast Pacific led him and his team to include this topic as their investigation. So in 2007, they founded the citizen science program Científicos de la Basura, litter scientists, which has since been expanded to other countries along the East Pacific. Welcome, Martin. Nice to have you.
Yes, thanks. Thanks a lot for the invitation. And let me try to share my screen. Okay, I don't know. Can you guys see my screen?
Yes, we can.
Excellent. Okay, so this is the title of the talk that you guys asked me for. And before I go into my talk, I would like to declare that I'm a marine biologist interested in all kinds of ecological aspects. And one of the things that we have been studying in our lab for a long time is these floating seaweeds and how they transport organisms around the oceans. And when we started this research, we also came across floating litter. And coming back to the shore, we realized, wow, there's a problem. But looking for information in Chile and in other South American countries for scientific information, we realized that basically nobody knew anything. There was no information. So then we continued for a number of years with our floating seaweeds, but at some point we realized we need to do something about this marine litter issue because no scientist, nobody was paying attention to that in Chile. And so we founded this program that is called Científicos de la Basura en las Playas. Means basically litter scientists. And with this project, we have done a number of studies together with school children from all over Chile, which is about 5,000 kilometers of coast. So it's from basically, if you are in Europe, from the North Cape to the north of Africa. So that's a spatial scale. And so we have generated scientific information. And if you are interested in this, you can go to this website and find it there. And since we are working with children, the questions we have been using or we have been trying to answer are the same questions as everybody is asking them, but we have been trying to make it as simple as possible. So for example, where does this litter come from? The question could be very complicated, the answer, to find the answer to that, but you could also make it very simple. Basically, is it from nearby or is it from somewhere else? So, for example, if you look at this picture, you can see there's litter on the beach that has no organisms growing on it. And then you have this bottle that has a number of marine organisms growing on it, and these organisms are large. So this bottle actually has come through the ocean with the currents and come from far away. Whereas the other litter that you can see on this picture has no organisms on it, so it has not come with the currents. It's most likely been deposited by users of the beach or has been thrown into the ocean very close by in the same bay and then thrown onto the beach by the waves. So this, of course, then organisms growing on litter is also a reason of concern to people because with this litter, the organisms can travel over very long distances. and those could be invasive species. And if you pick up some of this litter here, for example, this buoy open at the sea, you can sometimes find these invasive species. For example, this green alga here, that is one of the most invasive seaweeds all around the world, codium. And we also can find it attached to floating plastics. Then maybe you have heard about the litter that was released by the tsunami. And with this litter, a lot of organisms have crossed the North Pacific Ocean. Many species have been shown to travel on this litter and come from Japan to North America. So that's what's happening with large litter, with macroplastics. And another thing that's happening with macroplastics is, of course, entanglement that you have all heard about. And many different organisms are affected, fishes, birds, mammals, and also a number of invertebrates. But today we are not going to talk about the effects, the impact of macroplastics, which is, as we have just seen, species, transport, and entanglement, but we are focusing on microplastics. That's what you asked me for. And so microplastics, the main concern is ingestion. Organisms are ingesting these small plastic pieces, whether they are primary, as you have heard in the first talk, or secondary microplastics. The main concern is ingestion and everything that's related to that. And so the question that we're gonna focus on during the rest of my talk is whether ingestion of microplastics really is a problem. And as an overview, I want to cover these 4 questions. Who is ingesting? Why does it happen? Where does it happen? And is it really important? Do we have to worry about it? So let's first look at who is ingesting microplastics. Basically, you can say any organisms that you can imagine will ingest microplastics. It has been documented from fishes, from seabirds, from mammals. And nowadays, this figure is actually already 5 years old. And so now we know that also a lot of invertebrates like worms, mussels, corals, all kinds of organisms ingest microplastics. And we have heard about the sizes. So generally what people consider is that 5 millimeters and smaller is microplastics. And so you can see here on the scale here, larger things, larger plastics are ingested by larger organisms, and smaller stuff is ingested by smaller organisms. So if we look at 2 important groups here, for example, fishes and bivalves like mussels, you can see that large fish ingest larger microplastics, and small fish larvae ingest also smaller stuff. And mussels here, you can see, ingest a lot of very small stuff. And that's where it also becomes complicated analytically, because it's very hard to determine, to analyze this. And just as as a perspective, if you pick out now one of your hairs from your head and look how thick it is, it's about 0.05 millimeters thick. And there's still a lot of microplastics that's smaller. And those are the ones that are ingested by mussels and other bivalves like oysters, for example. And so why does it happen? And here we could go into a lot of detail. I will just show you one very simple slide. Here we are looking at a fish from Easter Island. And in this fish, we found these blue fragments, microplastic fragments, and we also found a lot of these blue copepods. And in fact, we found a lot more blue microplastics in the fish than we found in the surrounding waters. So these fish actually are selectively ingesting these microplastics, which are similar to their prey organisms, these copepods that you see here. And so microplastics are confounded with food. And that's what you see all throughout the animal, basically the different organism groups. They confound the plastics, the microplastics with their food. Also, it happens in many seabirds. And then, of course, there can be indirect ingestion of microplastics through their prey. We will come back to that later. So we have seen it happens in fish, also in seabirds, and also in mussels. We want to look at the mussels. And here is an overview study that shows a whole suite of different mussel species, for all of which it has been documented that they ingest microplastics. And here it happens in the same way as with the fish, as the seabird. They confound these microplastics with their food. And so we can say that there is, So there is a problem, and microplastics enter the food chain in many different levels. So where does this happen now? Let's look at that now. And here you can see a map that many of you may have seen before, where the highest concentrations of microplastics are. And this is in the open oceans, where the large oceanic gyres, basically the gyre currents, concentrate these microplastics in the open ocean. Now, the important thing is, where do organisms feed? Organisms feed where there is basically their food. And if we look at where the productive areas are in the world's oceans, you can see that actually these oceanic areas are actually very, very low productivity. And the highest productivity is in the coastal oceans. So in the open ocean, something that is happening everywhere is that currents concentrate stuff, all kinds of stuff. Currents concentrate plankton organisms, but currents may also concentrate microplastics and other plastics. So here you can see, for example, these lines where currents have generated these fronts. They are also called slicks. And so in this study, for example, these colleagues, they looked at these slicks where the currents concentrate stuff, as you can see here in this schematic figure. And so they took samples in these slick zones and outside of these slick zones. And you can see here on the right side, the light blue is the slick zones and the darker blue is the outside area. And you can see that in the slick zones, the plastic concentration is much higher than outside. The same happens actually with the larval fish. Inside, there's many more larval fish than outside these slick zones. So microplastics and these organisms are concentrated. And so that's actually then where these conflicts happen, where ingestion may happen. And here you can see in these frontal zones, everything is concentrated, the food and the plastics. And here is one thing that is very important, and that luckily also the former speaker emphasized. Once it is in the ocean, it is too late, because here the plastics in these areas where it is concentrated is intermingled with all kinds of organisms, and you can't take it out anymore, because you may actually do more damage to the ocean than if you leave it there. Because if you would take out the plastics, you would also take out all the organisms associated with these plastics. That's something very important to keep in mind. So here the plastics is interconnected, and here the risk of ingestion is specifically high, because to these areas is where the fish, the sea turtles, the seabirds, the mammals, everybody goes there to feed. And so that's where the risk of ingestion is very high. So now let's turn at the final and most important question. Do we really need to care? Is ingestion of microplastic a problem for all these organisms? So one question is that there may be trophic transfer through the food chain, so that the larger the organisms that are higher up in the food chain concentrate all these plastics in their body. And so that's one of the questions that is needs to be investigated. And so here, if you look at this figure, you see the trophic levels of different organisms and the amounts of microplastics. And you can see here higher trophic levels like predators. You see also higher numbers of microplastics per individual. And that could be because the microplastics accumulate in these organisms. However, these organisms of higher trophic levels typically are also larger. So then in this overview, this author actually concluded that looking at all the evidence, that there is no evidence that microplastics bioaccumulate in higher trophic levels. And so he suggested there is no trophic transfer through the food webs. And why should that be? Well, basically what is happening here, if you look at this schematic figure, again from Easter Island, you can see here flying fish that ingest sometimes microplastics. These flying fish are very important prey of tuna fish. And so the tuna fish ingest the microplastics indirectly through their prey, through the flying fish. But since the tuna fish is so large and the digestive system of the tuna is also so large, the small microplastics basically flush right through their gut system. So there is really no accumulation because it goes right through out again. So what are other negative impacts that could happen in this? And so here is an overview study that showed that actually there is many studies that document that there is no real effect. Then there's a few studies that show negative effects. Of course, the important thing here to keep in mind is there's no study that shows positive effects. Okay, so there is some negative effect, but it's not so widespread as you might assume. So here, why could that be? Here we are looking again at the mussels, and here what these folks have done, they have fed the mussels with microplastics and with natural particles, like sediment particles, and at different concentrations. And basically the mussels do not care whether they ingest natural or microplastic particles. So here, the effect of ingesting microplastics is very low because the mussels are used to typically ingest all kinds of stuff when they are filtering the water. Here in a similar study where the authors offered different concentrations, and they also didn't see any clear effect of higher concentrations of microplastics. So then the next question is actually that there may be contaminants that are absorbed to the microplastics. That's something that happens in the ocean and has been documented. And the concern is that when now these organisms ingest these contaminated microplastics that have other contaminants, that then these contaminants could desorb in the body of the organisms and contaminate the organism. That's here's a concern. And so there's contrasting evidence right now. As we have heard, we are just basically, there's still a lot of things that we don't really know. And that's really the main concern. So for example, this author suggested, based on his overview, that there is very little evidence that microplastics can be a vector for these other contaminants here, the persistent organic pollutants, to organisms. Here's another study that did show actually that microplastics do serve as a vehicle for some contaminants that show up in the body here of these juvenile zebrafish. And here's a similar study where they fed contaminated microplastics to the chicks of a seabird, and they found that here in those that obtained the contaminated or that ingested the contaminated microplastics, there is a concentration of these contaminants in tissues. Other authors again did something similar and found relatively limited effects. So here the word is not really out, the final word on whether or not this is an important effect. So I would like to ask all of you now, what do you think? Is ingestion of microplastics a problem? Yes or no?
That's a great question.
I'm not qualified really to take the final call on this.
Yeah,
but give me one more minute.
Yeah.
So you can try to answer that question in the chat if you like right now. But I would like to give you one more thing to answer this question. Microplastic is only one of many other stressors out there in the ocean. We have, for example, many organisms are facing increasing temperatures. Then we have ocean acidification, we have eutrophication. So there's a whole suite of different stressors out there, and we need to pay attention to that, because adding one more stressor may basically tip the balance in the one or the other direction. So I would say, well, yes, my answer would be a tentative yes. We do need to be concerned about it, even though we don't have any very clear evidence that microplastic for some of these organisms is a very serious problem. So what can we say finally, summarizing? They do cause multiple impacts. It may be species specific. Some species or some life stages may be more sensitive than others. It will be habitat dependent. And also we need to keep in mind that there's synergies with other stressors. So microplastics is one more stressor in a stressed ocean, and probably the least necessary of all these stressors that are out there. Thank you.
Thank you very much, Martin, for this fantastic presentation. I'm going to now give the word to Dr. Heather Leslie and Professor Dr. Raymond Pieters, who are going to talk about the effect on humans. So your question, is ingestion a problem or not? Maybe can be addressed here. And when you guys are ready, we'll do a short question and answer with all three of you in the end of this hour.
Let me see where I find my presentation.
We're going to do a double presentation. Ray is going to start, and I'll take over halfway.
Yep, I have to find mine, but I will in a minute.
Okay, I'll in the meantime just say that Professor Raymond Pieters is an expert in immunotoxicology or immunotoxic effects of substances on inflammatory diseases such as allergy. His work balances between biology and chemistry and is separately required to solve chemical safety issues. Together with Heather Leslie, he will lead a European Commission project, Polyrisk, to develop knowledge and approaches to assess human health risk of micro and nanoplastics. And
I'm a bit struggling. Yeah.
I'm sorry. Does Dr. Heather have the presentation as well? Maybe.
Yeah, she's.
I'm going to share both. I'm going to share both.
No, I got it.
Oh, you got it?
Okay, I can just say that Dr.
I got it.
Leslie is a leading expert in international microplastics research, and for almost 2 decades, she has been pioneering studies on how plastic and industrial chemicals can impact our ecosystem, food, human health, and society, and what can be done about it. Go ahead.
I'm a bit stuck now. Can you hear me?
Yes,
Yes.
we can hear you, but we cannot see the presentation.
I'm going to share the presentation.
No,
Or
my
no?
screen is a bit stuck, so I don't see anything now. I have to
We're losing you.
close a few
I think we're
things,
losing your
probably.
audio.
Let me check again because. Yeah,
Yeah, now we hear you well.
I have to leave and get back to you. Do you? I'm a bit stuck, so I have to.
Ray, why don't I just show your slides
Sorry.
and you can talk because we can hear you. Oh,
Maybe you can share the slides anyway, and we hope that he can log in again.
yeah. What would you like, me to do mine and then he'll do his in the opposite
Maybe-
order?
Yeah, maybe we can do that.
Okay. Need to be in Zoom, sorry. Okay. You can probably see this.
Yes, we can.
Okay. So Ray's going to talk about a new project that we have, which is quite exciting, and he'll talk a little bit more about the inflammation side. But we're a team of kind of chemistry and toxicology mixed.
Sorry.
Ray, I just started, so we're going to switch our roles.
Good.
Okay. Switch our order. So I'm going to talk to you a little bit about the human side of things. Are we a little bit plastic inside? Well, we've been talking about framing the plastic problem as a problem of the oceans, and for the last few years, we've really been doing our best to also show that it's we are a highly exposed species, and this is for the last few years been actually on the political radar. We can see here that what is this plastic stuff? It's a mixture actually of copolymers and the additives. This is what Martin was just talking about, and we've been studying these for decades. We know a lot about them, but what about these crumbs? A lot of these are substances of high concern, but these polymers or copolymers with different monomers, different building blocks, I call these basically the spaghettis and this the sauce. But we're kind of interested in what, if anything, are these little chunks of the noodles doing? And is it safe to breathe in and eat and digest plastic dust? The exposure to microplastic in the living environment, we know a lot more about the sea at the moment, but we have seen some early studies in the air, in food and drinking water. And it seems like everybody, when you look for microplastics, you can find them. Also, it wasn't a study done at our laboratory, but a study in human feces. And there's also plastics not only going in your body, but also going out. And this becomes a problem for sewage treatment plants. But of course, I should also say about all of these studies, they were really microparticles. So these were really measuring the largest possible particles. What we're interested in is what we call the bioavailable fraction. So when it gets into your body, the things that you don't poop out or pee out, but the ones that really reach your bloodstream and your tissues and your organs and get stuck there, where they might cause problems. And these are the smaller ones, the low micron and the nano, so smaller than a human hair. And I call this the analytical terra incognita. Here on the Lennox globe, here be dragons, the areas that you can't really fill in what's going on there. And we've been interested in this for many years in trying to get down to increase our analytical power to look at these places where the dragons are. And sometimes I say, this is not a field for cream puffs, because it's really scary sometimes when you keep trying and you can't make these invisible things visible. But we did find a way. Very recently at our university in Amsterdam, and some other labs around the world are working very hard on this. And we're trying to improve the quality so that we can now study the real world microplastic exposure in humans. We've started by looking at blood and placentas and amniotic fluid and some other matrices. The first study was published about placenta, not by our laboratory, unfortunately, but some laboratory in Italy. And what we now have the ability to do is measure both micro and nano-sized particles for the high production volume polymers. We have an ability to measure very sensitively because there isn't a whole lot of mass in a gram of your blood, but we are able to detect that. And we have what's very important and kind of boring for the outside world, but we need a lot of checks and balances, which we call quality assurance and quality control, to make sure that what we are measuring in a tissue is not something that got introduced during sampling or on the way to the lab or on the way to the measuring instrument. Because as you know, we live in a very plasticized world with our whole laboratory is basically built out of plastic. So we have to be very careful that our samples don't get dirtied with this plastic. If we find it in us, how bad is it? The two things we do is we look at concentrations that cause adverse effects, like chronic inflammation. Chronic inflammation is a sort of a theoretical way of looking at plastic particle toxicity, because it is something that happens when you're exposed to other kinds of particles, we know from air pollution studies. And then we want to see actually a really big difference with the concentrations measured inside humans, because if we are not very highly exposed, we won't reach the concentrations in our bodies that will cause adverse effects. Because of this relationship, the risk only comes when you're both exposed to something that's very hazardous. Just like the virus, if the virus is on the outside of your body, it's not measured inside your body, it's hardly able to cause any toxicity, and therefore you have no risk. But it's, of course, a virus is inherently, can be inherently toxic. So the basic particle toxicity hypothesis is that particulates in general can lead to this inflammation by various steps in a cell. And when I found out about chronic inflammation, and that's a prelude to a lot of chronic diseases, I really want to make sure that plastic particles are not causing chronic inflammation and possibly chronic disease. So we are really in a pioneering phase right now. It's still a lot of terra incognita out there, but I would really like to get to the bottom of this question. And this is basically my bigger question. Are contemporary plastics applications congruent with our best science? Is this the best we can do to protect environment and human health? And when I started out working on this, we were talking about the Anthropocene. And then about 10 years later, it turned into the Capitalocene, which was maybe more descriptive. And then almost a decade later, we had the Catulocene, or however you pronounce it, which sounds a lot better to me, where humans teach themselves to live in full and rich harmony with their fellow beings, as Donna Haraway put it. That made me really interested, in transdisciplinarity, a new intellectual space, and that's why I really applaud your project. You approach it with an intent to solve complex problems, and there's many different benefits which I see for both art and science, which I've listed here a little bit. Mostly, I would say freedom from paradigms is a really big one, because you really get stuck in your paradigms, especially nowadays, and especially in chemistry. It's such a cameral science where we are kind of cut off from feeling what we're thinking, and sometimes even cut off from thinking. Sorry to put it so negatively. And I wanted to share just a 3-minute little video of becoming a sentinel species. You probably can't see this. I forget how to get rid of this thing at the top. But it's a quick little video of 3 minutes showing a recent art science project that we did at the intersection of the nature, the sea, microplastics, and our immune system. And then after this, we will go on to the polyrisk project, which will be presented by Raymond Pieters. So now I have to turn this on.
I was fascinated with this concept of sentinel species. It's a term taken from environmental toxicology, which is using other species as indicators for how our environment is affected by pollution. They're kind of these canaries in the coal mines whose well-being and health condition can tell us something about the ecosystem that we're affecting.
We live in an environment that's really plasticized. All of our products are generally made of plastic, and a little bit of our products get inside of us. So we are all a little bit plastic too inside. So we are actually potentially a very highly exposed species. Slowly, we're starting to realize that we could also become a sentinel species. And in fact, aren't we all sentinel species?
Our project is a science fiction film about two imaginary researchers who want to become sentinel species. And they do so by introducing microplastics from the sea to their immune system. This work is then mixed together with an actual scientific experiment where we extract macrophages from my blood, which are the sort of first responders of the immune system, and feed them with microplastic particles that can be found in our everyday environment.
A macrophage will react against everything that is dangerous to the body. They are big eaters. And the question that we have is what happens when macrophages actually eat microplastics?
Seeing in front of your eyes this sort of microscopic battle between my macrophages and the microplastics was just really crazy.
Actually, you should not work with your own blood. There's a convention that we shouldn't do that. But I can understand that it has a meaning for an artist. This has created a number of questions. Do you imagine that your macrophages are actually outside of your body right now and interacting with microplastics away from your own self? I can imagine that this has a deep philosophical meaning for an artist like Sissel.
It just brings another layer of importance and interest and curiosity into this whole realm that we study from a reductionist and non-emotional point of view.
Heather and Juan, who are also, I think, special unicorn scientists, they already have artists' mindsets, so they're really going full on with speculating along with me and think a little bit further than what we can say is actual scientific knowledge right now. So I think we're all sort of like wanting each other's job a little bit, which is good.
Okay, that's it from my side. I'm gonna give the floor to Raymond Pieters.
Right. So hopefully it works now. Yeah, got it. Can you see my screen?
Yes.
Good. So I go to the beginning. So thank you, Heather.
It's very big though.
It's too big?
It looks, we don't see like we see a detail of the slide.
Now you should see it, right?
Yes.
Is it okay now?
Yeah.
Okay. So thank you, Heather, for taking over. I was here all afternoon, but something went wrong. So let me say something about our new project that Heather and I are heading, and that will start in a month from now. And actually it's dedicated to sort of understand how and if we are really exposed to nanoplastics, what could that do to our health? And you have seen probably this fish, and this fish in this picture is in Utrecht, where I work and live. And it alerted us, citizens of Utrecht, to become more aware of our problem. But the problem, as you heard, is even bigger than this. It's about micro and nanoplastics. And as you've heard from previous speakers, it may concern the particles, but also may concern the chemicals in the particles. So not only the the polymers, but also all these additives that are in there to make a plastic, like catalyst or plasticizers, etc. In addition, these plastics may serve as a vector for not only chemicals, as Martin alluded on, but also microbes. And the next slide shows you maybe what it means if you think about it. This is a few pictures of sort of macrophages that also were shown by Heather. And here you see 3 photographs of macrophage-like cells, dendritic cells that contain microplastics. This is a bead of 10 micrometers. These are beads of 1 micrometer, and these are beads of really, really tiny size, 200 nanometers. And if you look at this table here, you see that these are in the same size as a virus. If you think that our virus now, SARS-CoV virus, has this relative size of 60 to 100 nanometers, which is smaller maybe than these particles, but in the same range. And here you see these plastics in the cells. So these little dots, red dots, are plastics, and you can imagine that they do something in these cells. They maybe disturb the action, the way they act in your body. And also here you see a picture of cells from the intestine in culture, in vitro, and you see all these dots here laying on your epithelial layer. Well, this could just be something like it, but we know that this may lead to all kinds of effects. We know from particulate matter that it can induce inflammatory responses. And we also know from all kinds of studies that particulate matter, and maybe not only plastic, but all the matter that we inhale or eat, may contribute to long-term effects. For instance, allergy, if you think about respiratory exposure. And we also know that it is a really complex matter we work with, because these effects of these plastics, they depend on the physical, chemical, and biological characteristics. So it's a really complex matter to study and to get a grip on. And that's why we started this project, PolyRisk. And in this project, we really want to know the risk, and in toxicology or in the risk assessment, the risk is composed of hazard and exposure. So for assessing risk, we need to know more about the toxic effects, but also about the concentration at the target. So imagine the concentration in our blood, with our blood. We as humans are probably mostly exposed via the air, not so much via drinking water, although we don't know. But imagine that in the air, you have all these tire wears and all these pollutants in the air, and you cannot avoid exposure via the air. It goes in and it may never go out. Anyway, we don't know yet. So we have to assess the exposures via different routes. And that's what we, in our project, will do. Essentially, we will look at real human exposures and try to determine the chemical appearance, so the appearance of these micro and nanoplastics, any form, in our blood, and link that to possible immune responses. We will do a number of field studies with a number of groups over Europe. For field inhalation scenarios, we will do with indoor athletes in indoor soccer grounds, for instance, but also look at textile industry workers, and also do a drinking ingestion study. So just to know how and to what extent we are exposed, and whether that leads to an inflammatory response. So that's my short presentation. I just also want to stress that I really like to be involved in this, and I really admire that you students all will think about this. And this is a quote I found actually at the premises of the VU in Amsterdam, the Free University, so the University of Heather. And this I really like a lot. There's no science without fancy and no art without fact. And I hope that with your help, we and citizens become more aware of the problem. And for that, of course, we need an open mind. But also, I hope that in the future, with all the knowledge we have, we will get to a green toxicology view, and maybe circular by design. So think before you waste your product and use it well. So one solution I found, it just responds also to the presentation by Madhuri, there are some solutions. At least people think about solutions. Look at this, where they have made came up with a device. It's not yet in production, but to collect at least the damage of the particles that we release from our tires. And also there are some solutions, and that's, well, I still like to think in solutions for this problem. There's also ideas that we can use maybe fungi to get rid of our plastics. But above all, we need to make sure that we waste less, of course, and produce less plastics. So that's what I had to say.
Perfect. Thank you very much,
Thank you.
Raymond and Heather. I would ask Martin also to turn on his screen so we can have a little question and answers. I have to say that while Dr. Pieters was talking, he was also responding in the chat. Whether
That's
the
not me.
question...
That was me. I just changed
okay.
my name. I saw that I wrote that
Okay.
you don't have a split personality.
With the double tasking here, but okay. That question was answered. If you could drink microplastics, or you could absorb them there. We have another question that came in: is do you think that our economic infrastructure could easily function upon a world without plastic? And this is a bit more open-ended. See if any of you feel like addressing.
I addressed it. I think probably as I don't know if I was anonymous. Sorry, Ray. I was like,
yes.
I was saying, never underestimate human ingenuity. And if you want something, you can of course get it done. And but of course, impossible things will take a little while longer to get done. That's general rule. But of course, we are kind of locked in many ways to this technology that's biting us back, and that's why it's going to take a long time. But I always, if I was talking to high school kids and I said, "Can you imagine, my grandmother or great grandmother? She wouldn't believe that I was standing here in this auditorium in the university, a woman with a PhD." And by the way, I even could vote in the election and everything. And that's like unthinkable until it actually happens. And so I try to help myself realize that, it's really hard to imagine, but it's certainly possible. And I think our future will not have the same types of plastics in it as we have now.
No, but it's also about indeed the type of plastics. I mean, if you have your sewer from non-plastic material, it may not. How we use the plastic nowadays. I remember that my mother kept all the plastic bags. She never threw it away.
Raymond, we hear you
For a minute. I remember these plastic lids on your coffee cup. You throw it away.
I think it's better now. Martin wanting to say something, and then I have a question specifically for Martin. Go ahead.
Yeah, I wanted to say, I think the goal is not to live in a world without plastic. The goal should be to make better use of the plastic, because plastic is fantastic as a material, right? Yeah. And so the problem really is generated by the misuse. And I think also the plastic issue and a lot of other issues here in Latin America about social issues, the COVID situation, for us is an opportunity to rethink our way of life. And actually Raymond showed a picture with this car with like trying to capture these microplastics, which I personally think, no offense, is the wrong way of thinking. In fact, I think Raymond is living in the city that is the paradise of mobilization. Yeah, the problem is not the microplastics, the problem, or the tire wear, the problem is our way of life. And also with respect to all the biopolymers, where are they coming from? These supposed degradable plastics. It's just an issue of maintaining our way of life, throw away way of life. And that's the fundamental issue at the basis of everything. Everything in a way is connected to our way of life. And the plastic is just one of the symptoms of that. And so I would say Yes, of course, we should look for alternative products, better ways, better design, but also we should look and think about a different way of life. That's my.
I like my way of
Agree.
life right now, though, and I don't really want some eco dictatorship to say this should be your way of life, you know. And so I think it's a little. I would like to say that I think it's naive to say that in 100 years we're going to use the exact same materials as we're using today. And I think we'll probably innovate something in material science between now and a century from now. And so but I also like to be open to all of the, I think everybody should experiment with their idea of how to solve the problem, because there's not a silver bullet. We need a huge, sometimes I call it a big tree of ideas, and we have to scale up the things that are working. And we need transdisciplinary discussions, because we need to get out of our paradigms of thinking that, well, we just have to change the way we live. Maybe we do, but maybe we don't. Maybe there's other ways. I can still ride my bike around Amsterdam without releasing microplastics every time my wheels turn, you know?
I hope so.
So I don't want to stop biking, but I want a better kind of tire, you know?
Yeah.
So you always have friction, of course. So you always lose material.
Yeah.
And that's not a problem if it's within boundaries. I mean,
if it's a nutrient.
It's, well, for instance, if you think about the fungi that can eat plastics, who cares? You know? It's about the abundance that we produce. We produce so much that our system cannot really cope with it. So if we find a way to reduce the exposure or the appearance of plastics in what way or whatnot, and make it more circular. I mean, plastics are a product of nature, right? They are oil products. They originally come from plants. The oil is a plant-based material, but we sort of modify it in a way that it is not, well, we didn't think of the circular way of using plastics, which we should and have to, I think.
I have 2 or almost 3 questions, but we have to really wrap up, unfortunately, if we want to break before the next one. But one question posed by Katja is there evidence of microplastics contaminating fruits and vegetables? If so, does this elevate the impact on food change and therefore deepen the issue?
Yeah, well, it's a basic, well, Heather knows better, but it's basically present in all our food products except red wine.
Only drinking wine now.
No, but it's beer is also
I never tested red wine, just Chardonnay. But
yeah, someone did, I heard. So I've seen some data. Red wine is most clean, at least, but not sure. I
think especially processed food contains the most. This is my hypothesis. I haven't tested everything, right? But I know during production, you know, you have big containers where the tomato soup is being made, and there's what we do in order to keep those blades rotating is we spray little Teflon microplastics on the rotors. So and it's called food contact microplastics. You're allowed to put it in your tomato soup. And there's just many ways. We tested organic honey, you know, but there's loads of microplastics in honey because they're packaged and they're transported in great big plastic bins, and you know the air fallout. You know, you get a little bit of fallout in the air, and then so there's. The more processed the food is, I think the bigger the chance you'll have microplastics in it. I don't really worry myself too much about what's on the inside of an apple because of the, although that's been proposed lately that microplastics can be taken up in roots. I think we need to do more research before we really conclude that, because if you think of a plant with a cell wall, it's different than an animal cell. It has this cellulose crunchy outer layer, and the pores are so small that it's very hard to get anything except for the tiniest nanoplastic through those cell walls. So I think plants actually have an extra line of defense, but I'm not sure about it. But we have hardly any studies on that.
Thank you very much. There is one more question that I am going to ask, since Heather has been so good at the chat, if you could answer in the chat. So we can all get 5 minutes before the next session starts at 4 o'clock sharp, where we'll start with the first counterculture block. So we'll keep seeing you all there in 5 minutes. Thank you for your participation, all three of you. It's been wonderful to have you.
Thanks for having us.
Happy that we could have some of the interdisciplinary discussions there.